Hello everyone, and welcome to our launch webinar of the Torrent Genexus Dx Integrated Sequencer. My name is Erika Leishman, Regional Market Development Manager here in EMEA, and your host for today's webinar. Today's educational seminar is presented by Laboratories and brought to you by Thermo Fisher Scientific. We encourage you to participate in today by submitting any questions you may have during the presentation. To do so, simply type your questions into the Ask a Question box to the right and click Submit. We will answer as many questions as we have time at the end of our presentation. You may also have or want to use the Ask a Question box to let us know if you're having any trouble seeing or hearing the presentation. This webinar is educational and offers free continuing education credits. So please click on the continuing educational window at the bottom of the screen to obtain your credits. With this, we are very excited to introduce to you our speakers. First, we have Dr. Andy Felton, Vice President of Product Management and Market Development of our Clinical Sequencing Division. Andy will share with you how to generate NGS results in as little as 24 hours with the new Torrent Genexus Dx Integrated Sequencer, a fast, flexible, automated NGS CE-IVD system. This presentation will be followed by Dr. Luca Qualiata, Vice President of Medical Affairs for our division. Luca will share with you, present to you our mission in oncology, democratize NGS to improve access to personalized medicine. And finally, I'd like to introduce Kathy Davey, Vice President of Translational and Clinical Oncology in the Clinical Sequencing Division. Kathy will present to you our proven track record and future plans in delivering IVD for NGS. Before I'm handing over to my colleagues, again, I want to remind you, you can put any of your questions into the chat. With this, I'd like to thank you, enjoy the session, and I'm handing over to my colleague, Andy Felton.
Thank you for that introduction. We're really excited to talk to you today about our upcoming launch of the Genexus Dx system. As you see from this slide, we've really been getting ready for IVDR coming into force in the European market via the new medical device regulation and the in vitro diagnostic medical devices regulations. These regulations are really designed to create a new, robust, transparent regulatory framework that creates a fair market access for all manufacturers and for healthcare professionals. And this will enable both instruments and instrument consumables to be Class A CE marked products and meet the IVDR regulations by now, the date of May 26th, 2022. And these regulations are going to be implemented in all EU and EFTA member states, respectively. With respect to our introduction, we're really excited to bring the Genexus Dx Integrated Sequencer to market. The system was designed from the outset to be a diagnostic device, and this is the first introduction of the diagnostic version of the platform. The system's intended use, you'll see here, it's intended as an in vitro diagnostic medical device to be used by trained personnel for automated, qualitative, targeted sequencing of both human and DNA RNA from formalin-fixed tissue or from plasma samples. And the system consists of the Torrent Genexus Dx Integrated Sequencer and the Torrent Genexus Dx Software. As part of this introduction, we really have a whole suite of tools to help you with the transition, both from assistance, creating a personalized plan, or promotions that maybe you may be able to qualify for. And we're really committed to helping you build, and we are going to build and market IVD devices for the coming years. The Genexus Dx Integrated Sequencer builds off our Genexus Dx sequencer that we launched a couple of years ago. And the primary benefits are really the same. So fast turnaround time in as little as 24 hours. And that really helps to enable fast decision making for the tests that run on the platform. And a really highly simplified and automated workflow with a single touch point And that enables a really easy adoption by a broad spectrum of laboratories. With that automated, simple workflow comes improved lab efficiencies and ease in training personnel and reduced labor costs from people having less time in front of the instrument itself. Together with the informatics portion and the assay component, which really delivers the results with minimal tissue requirements with maximum success rates, it really is a seamless platform. And economically, it's implementable even for small numbers of samples, as little as one sample or many samples with the chip itself, and getting to generate results quickly when you need them. So as we outlined a little bit on the previous slide, it really removes some of the barriers to NGS adoption. The barriers around NGS adoption have been the time taken. In a lot of labs, it takes oftentimes multiple days up to 3 weeks in some cases to return results. The expertise required to run the platforms typically has required multiple instruments with multiple touch points across multiple suppliers. And that's meant that training and hiring costs are very high for lab technicians. And also the cost to run small samples, batches to run quickly has been too high. And similarly, there's a problem around the amount of tissue in most cases that's available to be tested. And in this case, our technology is allowed very limited amounts of tissue, and that really helps us avoid quantity not sufficient failures. So really the advantages of the systems are it returns the results quickly, it's simple, it's flexible, and with as little as 10 nanograms of input nucleic acid, generates results with a high sample success rate. And that really removes a lot of the major barriers to implementing NGS in a variety of laboratories. So let's have a little bit of a look at the instrument itself. This is the Genexus Dx Integrated Sequencer. You can see on the top left, we have a vision system, and that helps the users place the consumables in the correct place. And it also identifies that the correct consumable is in the right position via the touchscreen GUI you see below. The consumables are very simple to place. There's a series of plastic single-use disposable consumables as tips that we place into the instrument. It takes about 5 to 10 minutes of setup time, guided by the touchscreen GUI. And then post-run, there's a UV light that allows for decontaminating the instrument. So very simple to set up and easy to run. The process includes a variety of the steps that are involved in next-generation sequencing, including library prep, and that includes the cDNA synthesis steps for samples derived from RNA, the clonal amplification or templating step, sequencing, the primary data analysis and variant reporting, both for FFPE DNA and RNA, and for cell-free total nucleic acid applications from plasma. Up front of the Genexus Dx Integrated Sequencer, you can choose to use the Genexus Purification RUO platform, and we are working on a program to take that platform to IVD. At the back end of the system, we'll output a report that is simple to review and analyze, and gives the data in a very condensed or more expanded form, depending on how you need to deploy that to your partners. On the Genexus Dx Integrated Sequencer, you can prepare and run up to 4 different assays simultaneously in a single run. Those assays can be run with 1 or 2 different chemistries, the AmpliSeq chemistry or the AmpliSeq HD chemistry. All 4 assays must be of the same chemistry and broadly should have a similar thermal profile to be executed within a single run. The automated library preparation capability is capable of delivering up to 32 library preps in a single run. The number of library preps depends on the configuration of the exact assay that you wish to use. So in the case of a single pool assay, that would be 32 samples. If the assay consists of 2 pools of amplicons, that would be 16 samples at a time, and so on. If a 4-pool assay is used, it would be 8. One of the major advantages of the system is up to 2 weeks of on-instrument stability for the chip and reagents. The chip is a multi-lane chip, and lanes can be addressed independently. So you can use 1 or 2 or 3 or the whole chip's lanes in a single go. And if you don't complete using the whole chip, the chip remains on the instrument until the next time the samples are ready to be run on the system. The assay and throughput flexibility is really facilitated by strips and this multi-line sequencing chip. So the number of strips really equals the number of samples, and you place the correct number of strips guided by the GUI on the instrument. In implementing the solution in your lab, we have a variety of services to help you get the system up and running, including global customer concierge. They help you prepare your lab space. They can connect with your IT department to assure that network connections, including advantages from our cutting-edge digital support options. We obviously have trained and qualified field service engineers, and we have packages that include our field application scientists or clinical applications consulting groups to get you up and running quickly and easy on the assay of your choice. Once you're running, we also have qualification services, including IQ/OQ and PQ services for the systems available, as well as standard service offerings. Let's have a quick look at the Genexus Dx Integrated Sequencer features. In particular, the software. The Genexus Dx has a different software, as you might expect, and within that software there are 2 modes. The IVD mode, which will be for use with the IVD assays, and then an assay development mode, which allows the lab to run a variety of assays for clinical research purposes, so research level assays that run on the platform. And you select which mode you wish to operate in at the beginning of the process. The run setup in either dual mode software is pretty easy and intuitive. It's about a 5-step process. The results are organized into tabs that allow you to interpret them with minimal bioinformatics experience. And the reporting on the variants is easily customizable and can be connected to our e-signature support to allow for approval of reporting through a chain of custody. Data can be automatically input or output from the system via some APIs. So if you're using laboratory information management systems, you can connect to those APIs or connect third-party softwares for additional analysis tools via those automated outputs. A little more on the data flow. You can see on the right-hand side of the slide that we have a number of pre-packaged workflows, and they vary on the actual starting point in the process. So you can see from nucleic acid to result. So if you have isolated nucleic acid, you can start there. If you have prepared libraries, you can also start at that position. And you also can take data, raw data in the form of BAMs to the result as well. So there's multiple places you can start. Those prepackaged workflows run on either the assay development. You can see the little key there, the dark blue and purple shaded boxes. If they say AD and IVD, both those capabilities are available in assay development and IVD mode. There's a dashboard highlighting recent runs and run status for in process, as well as QC checks on inline controls, as well as the instrument status. Within assay development mode, you have the capability to create custom assays as well. And as I discussed earlier, you can connect the system to LIMS. You can also easily enter sample metadata via the tools or the APIs and connect to our cloud, Thermo Fisher Cloud Connect platform, or other Genexus servers in assay development mode. In terms of variant reporting, once the data has been produced, we have a key findings tab that summarizes the key metrics and quality control and the coverage per gene. So some of the things that you should be looking at on a per run basis to review the quality of the run and the run metrics, as well as a variant tab that lists the complete sets of variants called, as well as the annotations. and allows you to filter those variants by some customizable filter options. There's also the ability to visualize the data at the genomic level with an interactive tool, and then customizable reporting via the report tab using pre-built or user-defined report templates. So very easy processes to manage and look through your data, including the ability to back up data and restore locally, as well as save data to your external storage mechanisms or to an AWS cloud account. One of the important features for a diagnostic level instrument or a diagnostic labeled instrument is the ability to be compliant with 21 CFR Part 11 and Annex 11 guidelines. And the Genexus Dx software contains all of those. So we have both audit tracking, internal security, access control, data backup and restore, and electronic sign-off of data. So you can see some of the features here. So audit tracking that generates a secure timestamp audit that records the date, the reason for any change in the data, and this also tracks and prevents users from being able to modify or delete audit trails. Allows for sign-off of electronic records, including the names, dates, and times, the reason for signing. And obviously we have security, including data encryption and the ability to update the software with security patches. So full 21 CFR 11 compliance support. So how do you get access to a Genexus Dx Integrated Sequencer? There are really two options. You can purchase the Genexus Dx system that's available now and will be shipping from the end of March. Or you can upgrade your existing Genexus RUO instrument to IVD slightly later this year. And with that upgrade, no hardware changes are required. We simply come in, update your software, and update the labels on the system to comply with the regulation. And the entire process is done with a field service specialist visit. So if you wish to do that, you can access the DX capability by upgrading your existing RUO instrument. So with the launch of the Genexus Dx system, we're really driving towards a complete end-to-end CE-IVD marked NGS solution, helping to make NGS accessible for everyone. Starting with the patient sample, extracting the nucleic acid with the upcoming Genexus Dx purification system that will be coming later this year, and then moving to the full next generation sequencing workflow with Touchpoint 2 on the Genexus Dx Integrated Sequencer that's now launched, and then developing the final Genexus Dx reporting with the software tools that will bring the Dx report from the IVD assays that will be coming soon as well. And with that, I'll pass it to my colleague from Medical Affairs, Dr. Luca Quagliata.
Hello everyone, speaking is Luca Quagliata. A warm welcome to all of you joining us today for the official launch event of the Genexus Dx Integrated Sequencer. And thank you very much, Andy, for setting up the stage with your presentation. For what concerns my part, I would like to focus on the clinical implication and impact that a unique solution such as the Genexus Dx Integrated Sequencer will have in the field of oncology. Designed for use in clinical laboratories, this fully automated and IVD compliant system enables users to perform both diagnostic testing and clinical research on a single instrument. And there will be no better start for me than talking about the overall aim of precision medicine, a pretty straightforward goal to improve patients' outcome. And that is perhaps something that has its most pivotal example manifested in the area of lung cancer, and more specifically non-small cell lung cancer, where a wide range of highly effective approved targeted therapies exist and continue to grow, being directly associated with specific oncogenic drivers and biomarkers requiring accurate and fast companion diagnostic testing, as clearly shown in this slide and supported by a significant amount of data, companion diagnostic testing has effectively improved overall survival for many non-small cell lung cancer patients. And now, thinking about it, precision medicine is truly a biomarker-driven story. So it doesn't come with any surprise to look at the last 20 years of clinical trials in oncology and notice, as displayed on the left side of this slide, that the number of biomarker-driven trials has simply skyrocketed. We now more than 60% of all clinical trials in oncology incorporating biomarkers for patient selection, and importantly, all this has translated into a massive increase in the number of personalized medicine now available on the market. And perhaps it is in this slide that the best representation of how the future might look like is found. With the number of both tumor-agnostic and tumor-specific therapies continuing to grow and fast, it is thus clear that in this context, testing single genes simply cannot keep up the pace. Another critical factor that will dictate the true impact of any meaningful molecular profiling solution is testing turnaround time. Indeed, delivering multiple biomarker testing by means of NGS can only exert its full clinical impact when it is timely performed. In this slide, I'm sharing some of the data presented at the latest annual AMP meeting, whereby scanning electronic health records of non-small cell lung cancer patients, over 1,000 of them, with the help of our collaborators, we observed that the average turnaround time for NGS tumor biomarker testing is about 25 days in the United States. As a direct consequence of this, in this dataset, roughly 30% of cancer patients were treated irrespectively of their molecular profile result. In about a third of them, which were originally exposed to either chemotherapy, immunotherapy, or a combination of them, needing to switch to a targeted therapy after the NGS results were eventually available. Overall, this data highlights how important it is to get results fast and closer to the patient. Lastly, while these results have been observed in the United States, we are equally collecting similar data across Europe, and we'll be sharing a full data set in a number of upcoming major medical meetings. Despite the fact that a substantial number of evidence suggests that multiple biomarker testing is the most meaningful approach for certain tumor indications, so many laboratories around the world still do not use NGS. It is part of our mission to improve all this and to democratize access to NGS, as we truly believe in its value for personalized medicine. And we certainly are not alone in believing this. We have done a series of interviews with pathologists, laboratory directors, and oncologists from several countries across the globe. And this is what they all agreed on. Significantly shorter time to result enables faster and optimal treatment decision. Small sample requirement provides biopsy stewardship, tissue saving. Local testing improves care coordinations between multidisciplinary teams, leading to a true personalized medicine, along with enabling the growth of local expertise in biomarker testing to support not only the present, but also the future of precision medicine. And now, I would like to conclude by saying that with faster answer and NGS reports in as little as 24 hours, the results generated with the Genexus Dx Integrated Sequencer will greatly aid clinicians and caretakers in their patient management decision process. Thank you everyone for participating in this event today.
You just heard from my colleague about how precision medicine has been transforming cancer patient care and the importance of bringing genomic testing closer to the patient in order to deliver the results faster and help improve clinical outcomes. Thermo Fisher Scientific has been a leader in enabling this transformation with its portfolio of NGS Companion Diagnostic solutions. We were the first and currently the only diagnostic company to offer distributable NGS Companion Diagnostic assays. Our current NGS IVD assay is now approved and reimbursed by government and commercial payers in 17 countries worldwide. In collaboration with several regional and global pharmaceutical partners, we now have 14 approved companion diagnostic claims to support 14 approved targeted therapies. This growing list of companion diagnostic claims with our Oncomine solutions is enabling clinicians to use the latest diagnostic tools to better manage their patients with available targeted treatments. The Oncomine Dx Target Test is the only distributed NGS IVD kit with companion diagnostic claims. This assay delivers multiple biomarker results for multiple targeted therapies in non-small cell lung cancer and cholangiocarcinoma from a single tissue sample. As the first NGS companion diagnostic test to receive FDA approval, it has subsequently expanded globally to Japan, Korea, the EU countries, and is now registered, reimbursed, and commercially available in 17 countries. We are expanding global access to the Oncomine Dx Target Test with registrations planned for multiple countries in the coming months. Thermo Fisher Scientific's track record of delivering NGS IVD assays dates back to 2015, when we launched the first Oncomine IVD kit with profiling claims for solid tumors. As noted in the previous slide, we launched the first NGS CDX test with Oncomine Dx Target Test back in 2017, and those CDX claims have now grown to 14 claims and will continue to expand in the future with our growing pharma collaborations. We are very excited about our latest NGS IVD assays scheduled to be launched later this year. This will be our first NGS CE-IVD assay that will operate on our latest NGS platform, the Genexus Dx Integrated Sequencer. This assay is called the Oncomine Dx Express Test. This pan-cancer assay is expected to generate new CDX claims in the coming years as we collaborate with pharma partners in broadening access to NGS diagnostic testing worldwide to support their targeted therapies. The Oncomine Dx Express Test will launch as a CE-IVD test soon and will operate on the simple-to-use and rapid Genexus system capable of delivering comprehensive biomarker profiling results in as little as 1 day. This 46-gene pan-cancer assay will simultaneously test for relevant variant types important in cancer, including insertions, deletions, substitutions, copy number gain, and fusions. With a single test and simple workflow, the assay can be used with both FFPE tumor tissue or liquid biopsy samples, maximizing the number of patient tumors that can be profiled. Using our latest AmpliSeq HD-based technology, this assay includes molecular tagging to enhance low-level variant detection, which is particularly important for detecting cell-free DNA and RNA in liquid biopsy samples. The assay will also incorporate fusion sync detection technology, delivering high sensitivity and specificity for detection of known and novel fusions. And finally, based on ESMO guidelines, the Oncomine Dx Express test will detect 100% of the most clinically relevant biomarkers in non-small cell lung cancer, in addition to significant coverage in other tumor types. The Oncomine Dx Express test on the Genexus system will remove major barriers for labs to incorporate NGS in-house for clinical diagnostics, bringing precision oncology close to the patient. Using as little as 10 nanograms sample input maximizes sample success rate. The workflow requires only 2 user touch points and 20 minutes of hands-on time, delivering results in as little as 1 day, whereas other NGS technologies could take several days or even weeks when samples are sent to outside labs. With such fast turnaround time, results can also be combined with other fast techniques such as IHC to enable clinicians to make the best decisions for their patients without delay. The Genexus system offers the only true end-to-end integrated NGS workflow. This chart lays out our Genexus CE-IVD roadmap for the year. Following the launch of the Genexus Dx Integrated Sequencer this month, we will launch 2 more CE-IVD solutions by the second quarter. This includes the Oncomine Dx Express test, which was described earlier. With the Express test, we will also launch the Oncomine Reporter Dx, an integrated, easy-to-use genomic analysis software to explain NGS data and enable streamlined access to a final report with annotated sample-specific variants. Later in the year, we will launch the Genexus Dx Purification Instrument to provide a fully integrated and complete IVD workflow. Thermo Fisher Scientific is committed to delivering additional clinically validated assays that will operate on the Genexus Dx in the future. These assays include, but are not limited to, hemato-oncology, comprehensive genomic profiling, women's reproductive health, and infectious disease, among others. Further details regarding these assays will be made available in the near future. Our plans for the Genexus IVD end-to-end solutions go beyond countries accepting CE-IVD. We have plans to submit for registrations in the US, Japan, and other countries. The Oncomine Dx Express test, which will be launched later this year, will also be compliant with the new IVDR directive in the future. With our pharma partners, we plan to expand claims beyond profiling to include companion diagnostic claims. Where possible, Thermo Fisher Scientific proactively seeks to collaborate with industry partners to expand NGS IVD solutions to labs. To demonstrate our commitment, we recently announced an agreement with Oncocyte Corporation to develop and co-market the Oncomine Comprehensive Assay Plus on an IVD assay on the Genexus system. In the future, labs will be able to use this 500-gene panel for clinical diagnostics. The agreement also includes clinical validation of Oncocyte's Determa-IO, a 27-gene expression assay with the potential of a broad range of immune oncology clinical diagnostic applications. The agreement with Oncocyte will help pave the way for IVD clearance of these assays for tumor profiling and future use as companion diagnostics. We are on a mission to democratize NGS to support patient access to personalized medicines. Thermo Fisher Scientific is committed to partnering with you in your IVDR transition. The Genexus Dx Integrated Sequencer is now CE-IVD marked and available for purchase. Join us for a webinar on the 26th of May to learn more about the Oncomine Dx Express test on the Genexus Dx.